I will just add a comment to that. I think the biggest challenge, or one of the biggest challenges in psychedelics becoming medicine, is dosage. Yeah, so how do you dose it? At the moment, our best guess is body weight, but this is just as vague as it can be or variable. One has to take in care like metabolism and many other factors, and probably that would enable us in the future to be able to also handle cases which are a bit more sensitive, because we want to treat mental health disorders, but many people are excluded because they have pre-existing conditions from studies. Okay, let's move on to the next talk. So it will be Daniela. She is doing her PhD in France. We met at a psychedelic Frontiers in Psychedelic conference in France this year, and she agreed to present talk about preclinical studies of LSD, and I will put the talk on. Yeah, thanks, Daniela. So the floor is yours.
Thank you, Brett. Thank you, Bradley. Guys, this is the last boring talk before the breathing workshop, so bear with me. My goal is to give you an overview of what we know in the preclinical field, and we will go through receptors, we will go through neuroplasticity, circuits, and behavior. So even if you get lost in one of the parts, you can still follow for the rest of the parts. So as Pradeep said, thank you. We met in a conference. I'm doing my PhD at the Paris Brain Institute at the lab of Eric Bourguier. And I decided to call this talk The Curious Case of LSD, a Preclinical Perspective. And this is in an allusion to the movie The Curious Case of Benjamin Button, where Brad Pitt is born an old man and then he dies as a baby. And I think, among others, researchers, but also this happens in the psychedelic field and with LSD in particular, right? We have followed this similar reverse scientific path where we have more information on the clinical outcomes than on the preclinical side. We know more what's happening when we give LSD to a patient than how is it function. So, and why is that? So, let me briefly introduce you to what I call the Benjamin Button timeline of LSD. It's going to be very brief. So due to the lack of legislation on drug prescription, it took only 4 years for the first clinical trial on LSD to be published after the first experience ever by Albert Hofmann in 1943, in comparison to more than 10 years for the first study in mice to be published. And back in the time, in the 50s, LSD was being widely distributed to neuroscientists and to researchers, psychiatrists, for investigational purpose, which led then more than 40,000 people to be administered between 1950 and 1965. And I think we all know the rest of the story. So today I aim to give you an overview of the advantages and limitations of using animal research in the field of psychedelics. And I will mainly focus on LSD because it's the subject of my thesis and also of this talk now. So before we dive in, I want to tell you some of the things, some of the difference also that are important to take into consideration when comparing LSD studies in humans and in rodents. So, for example, the LSD interaction with the human 5-HT2A receptor is characterized by much lower kinetics in rodents. In terms of the drug itself, humans are usually given the hydrophilic salt version orally and only once, whereas in animal research, LSD is injected intraperitoneally, sometimes for up to a week. We are not confounded by expectations about the therapeutic effect in rodents. There is no placebo effect. And more importantly, we can do causal studies and analyze the mechanism of action of LSD in depth. But what do we really know and not know in the preclinical field about how LSD modulates the brain at the molecular, neural, and behavioral levels, both acutely and, more importantly, at the long term? We know that the first molecular targets of psychedelics are the 5-HT2A receptors. And we know this because acute effects both in animal models and humans, they can be prevented by either pharmacological blockage with 5-HT2A receptor antagonists like cantacerin, or deletion of the 5-HT2A receptor gene, as in knockout mouse studies. And moreover, there are even studies that show that the percentage of occupancy of these receptors can serve as a predictable measurement of the intensity of the subjective effects in humans. What do we not know is which specific pathway or pathways or even receptors are the key main responsibles for the long-term outcomes. At the 5-HT2A receptor level, the formation of heterodimers, the ratio between different receptors, their location, the cellular context, all of these factors are going to induce biased conformational changes in the receptors that in turn are linked to the activation of specific downstream signaling pathways and interactions with different proteins. And here I'm just showing you a simplified view of some of the biochemical pathways activated by psychedelics, namely the GQ pathway and the beta-arrestin pathways, and a small subset of the proteins 5-HT2A receptor interact with at the spine level. But the overall picture is much more complicated. And there are other main players implicated, like AMPA, TRKB receptors, that seem to actually be important to sustain the effects. And I'm not going to go into the details of any of these, and I know this scheme seems too complicated. That's the idea, okay? The idea is really to show you that the bigger picture is involving much more than the simple implication of the 5-HT2A receptors. On top of that, LSD in particular, when compared to other psychedelic drugs, has a very complex polypharmacology. It has been shown that in addition to having agonist activity at 13 of the 15 human serotonergic receptors, it interacts also with dopaminergic, adrenergic, muscarine, histamine, trace amine-associated receptors. So with this in mind, researchers are now developing strategies by either creating new compounds that are biased to a specific pathway, analyzing transcriptomic data, doing simulations, all of this to try to understand what are the mechanisms of action of psychedelics. And I'm going to point out 3 papers as examples. Earlier this year, it was shown by Vargas and colleagues from the Olsen lab that LSD and psychedelics in general activate 5-HT2A receptors at the intracellular level. From the Brian Roth lab, we became closer to knowing what is the preferential conformational change of 5-HT2A receptors to accommodate LSD, why it results in this exceptionally slow dissociation rate, and which pathway gets biased to. And it seems to be the beta-arrestin pathway. And the last example comes from Romanardo, postdoc at the Guldulan lab, that showed with transcriptomic analysis that psychedelics induce remodeling of the extracellular matrix. So we are starting to get more and more pieces of this, of what's happening, but it's still not enough to construct the entire puzzle. Another well-described phenomenon induced by psychedelics is neuroplasticity. And neuroplasticity can be investigated at multiple levels, from modifications of genes and protein expression up to behavioral changes. On the preclinical side, there have been multiple studies on psychedelics that show an increase in the expression of genes related to synaptic plasticity, synaptic strength, and dendritic growth after a single administration of psychedelics. And here I'm just showing you 2 examples of how a single dose of LSD increased the expression of immediate early genes, which are proxies of neuronal activity. At the cellular level, research on DOI was the first to demonstrate an increase in dendritic spine size and density. And later, similar experiments were done with classic psychedelics, which also revealed an increased dendritic arbor complexity, promoted dendritic spine growth, and stimulated synapse formation. And I hope you can all see with the naked eye that, for example, the cortical neuron that was treated with LSD has more dendrites than the one treated with vehicle. And One could argue that these modifications are merely structural and do not necessarily represent functional changes, which is true. However, animal studies in vivo have also shown that psychedelics can catalyze bursts of synaptic and dendritic growth, augment the AMPA-NMDA ratio, and increase the strength of long-term potentiation. And these are all indicators of functional neuronal plasticity. And here I'm just showing examples where this was demonstrated in psilocybin, DOI, and of course LSD. So there is a consensus in the field that psychedelics are psychoplastogens, that they induce neuroplasticity. But there are still some questions that remain. Is neuroplasticity dose-dependent? Meaning, are there different receptor occupancy that induce plasticity and hallucinogenic effect versus plasticity and therapeutic effect? Is there a critical window for neuroplasticity? Is there a limit period of time where the brain networks can actually adapt to these extrinsic and intrinsic signals? And if so, can we measure it in humans? And are there possible consequences of psychedelic enhanced neuroplasticity for both patient groups and healthy people? All of these questions actually require more investigations, but scientists also love to make hypotheses. So just 3 months ago, researchers from Johns Hopkins pointed out a correlation, and more precisely a proportionality, between the duration of the acute subjective effects in humans and the duration of the mice social reward critical period that stressed the potential importance of post-treatment integration. Moreover, they proposed a new working model where psychedelics act as a diverse array of binding targets. They trigger specific downstream signaling responses that lead to activity-dependent remodulation of the extracellular matrix, which is in turn this permissive event that is going to enable metaplasticity to happen. And in a nutshell, metaplasticity entails the changes in the physiological and biochemical state of neurons that alter their ability to generate synaptic plasticity. In simple terms, is basically the plasticity of synaptic plasticity. So again, the picture is much more complicated than at first sight. But tackling these questions with multiple approaches, in this case transcriptomics, can lead us to better understand the mechanism of action of psychedelics. At the circuit level, studies in humans have been consistently showing that psychedelics lead the brain to a hyperconnected state, and more specifically, that they decrease within network, but increase between network functional connectivity. And the most iconic representations of this hyperconnected brain state are these connectivity maps. So the ones on the left represent connected brain regions after administration of vehicle or psilocybin, and the one on the right represents already a subtraction between the connectivity map of LSD and control, with the red lines representing an increase in connectivity after LSD administration. In preclinical research, quantification of immediate early genes as proxies of neuronal data became a standard to study whole brain approaches. And the biggest advantage of this technique is that we are not limited to the cortex, but we can also have information of all the structures, deep brain structures included. And here I'm showing you a paper published this year from Alex Kwan Lab that compared brain regions that were targeted for ketamine and psilocybin, and showed, among other results, that the reticular nucleus of the thalamus, the PAG, and the caudoputamen were more selectively targeted by psilocybin than ketamine. There is also currently a paper on bioRxiv that is using the same techniques to address which common and distinct brain regions are modulated by psychedelics and its context. So this is just an example of one of the questions that we can easily address in the preclinical field that is more difficult to address in humans. Another important finding is that both in humans and rodents, psychedelic effects seem to be dose-dependent. So The intensity of the subjective effects reported by humans after psychedelics seem to increase with dose. And here, we already saw this figure today, but here is a comparison of 3 human studies on LSD, and hopefully we can all agree that the study that is representing the higher dose, 200 micrograms of LSD, that is this bluish purple line, is the one where people reported more intense experience. On the preclinical side, the Gregorian colleagues from the Gobi lab reported no changes in the firing of dopaminergic VTA neurons at low doses, but a substantial decrease at higher doses, suggesting then that dopaminergic pathways might only be activated when a certain dose is reached. And all of these results are super interesting, but the ultimate question we still don't have an answer is, are these neuronal circuit modifications persisting over time? Are they also dose dependent? And from the data we have, mainly data at the acute level, there are 4 main psychedelic network models that explain, at least in part, the mechanism of actions of psychedelics. And I'm going to go very briefly through them, just for you to know. So we have the cortical striatal thalamic cortical network. These models focus on the fact that we have altered thalamic gating and subsequent changes in sensory versus association processing. We have the Rebus model that stands for relaxed beliefs under psychedelics, that defends that idea that we have an increase in bottom-up signaling and reduced top-down predictions. The strong prior model, which suggests that psychedelic experience arises from a reduction of bottom-up sensory inputs and aberrant reliance on top-down expectations. And finally, the cortico-claustrocortical model that centers on disrupt claustrum activity and how it might lead to a desynchronization of cortical networks. So these models are not necessarily incompatible with each other, but they are still like a good representation of what are the questions that we still have in the field. Finally, in terms of behavior, so we did receptor, neuroplasticity, neurocircuits. This is the last part. Psychedelics became widely famous due to the acute effects they evoke on humans, which include but are not limited to hallucinations. And nowadays, beside the focus being on the sustained therapeutical outcomes, we still measure these acute effects, and we are doing it now in more controlled environments like the lab or the hospital. So we can evaluate these acute effects, and most of the time this is done through the data questionnaires where the participants' answers are rated on scales, such as the 5-dimensional altered state of consciousness. And the weird fun fact, it's actually 11-dimensional scale, you can count. So in animals, one of the most well-described and quantified acute behaviors is called a twitch response. So here I'm showing you a video from the Alex Kwan lab of a mouse that got administration of psilocybin, and I hope you can see these rapid side-to-side rotational head movement. And this occurs in rodents after the administration of serotonergic 5-HT2A agonists that induce hallucinations in humans. So in animals, we also have a proxy of the hallucinogenic component of the compound. And more importantly, Alberstadt and colleagues showed that one of the most well-described, Alberstadt and colleagues showed a strong positive correlation between the number of head twitch responses in mice and the reported hallucinogenic potencies of different drugs in humans. At the long-term level, the most consistent findings on sustained effects on animals after LSD administration have demonstrated an increase in anxiolytic, antidepressive-like, and pro-social behaviors. And I'm happy to discuss details of any of these papers later. So it seems that as in humans, even if not at the same time scale, we can observe in animals psychedelics triggering particular behaviors and sustaining these behaviors over a certain period of time. However, one question still remains. Are the subjective effects of psychedelics necessary for their enduring therapeutic effects? And from one side, clinical researchers have demonstrated strong correlations between acute experience and therapeutic response, suggesting even a predictive potential on the efficacy of treatment. On the other side, we have preclinical researchers that have been developing non-hallucinogenic compounds, so compounds that are not going to produce this at-twitch response, and that they still promote neuroplasticity and they still have like beneficial behavioral changes. And I'm going to point you out 3 examples. TBG, that was inspired in ibogaine, is a non-hallucinogenic compound with anti-additive properties. R70 is a structural optimized version of 5-HT2A receptor agonist that is a non-hallucinogenic but holds antidepressive activity over time. And the last example is the 2-bromo-LSD, a non-hallucinogenic analog that reverses the negative behavioral effects induced by chronic stress. So these results put into question the importance of the psychedelic experience for the long-term beneficial outcomes, and also became crucial for the development of new non-hallucinogenic compounds for human use. Of course, we don't know if it's the same in humans. So, in summary, I gave you a quick overview on what we know in the preclinical field of psychedelics, what are some of the crucial questions we still need to answer in the field, and I hope I could stress out the importance of combining efforts both in the preclinical and clinical studies to fully understand the potential of psychedelics to modulate the brain. I'm super happy to be here today with you at Insight to discuss more on how we can rethink psychedelics. And I want to finish by thanking my team, to thanking all the researchers I briefly mentioned through the papers I presented, all the others that I didn't have the time to do so, to thank all the amazing speakers in this panel, of course, Pratip for inviting me and organizing, and all of you for listening. And I leave you with 2 representations from Dali on the sentence, "A woman neuroscientist standing on the shoulders of giants," in a psychedelic style. Thank you.
So questions for Daniela? Maybe start here.
Thank you for the talk, very interesting. You have mentioned that paper about intracellular 5-HT2A receptors that have become like so hot topic last year. And I wanted to know your opinion because you also know that extracellular signaling of psychedelics is already very messy and very complex. We've got this bias agonism on extracellular receptors, and the intracellular cascades that are activated by either hallucinogenic or non-hallucinogenic 5-HT2A agonists is different at these receptors. Then you have this other view, this recent discovery, that you have another difference between hallucinogenic and non-hallucinogenic 5-HT2A agonists, that is this capacity to cross the plasmic membrane to activate the intracellular receptors. So what would you think it would be like because what could be more related like to the psychoactive properties of psychedelics? This extracellular bias agonism of membrane receptor in the plasmatic cytoplasmic membrane, or this capacity to get in and activate intracellular receptors? I don't know if maybe it's too broad or...
No. Thank you for your question. It's a lot, but I will try to give you my most informed opinion. So regarding the paper, usually on the preclinical field, one of the things that you will listen the most is data is data. So if you go and, of course, one of the problems on the preclinical field is also that all the papers almost that claim something are n equals to 1. So it's very difficult to make a lot of conclusions on one paper only. So we need to have replication of these results. However, this is a really neat experiment that they did. So basically, in this experiment, they could even show that, okay, if this is true, if it's really we need the hallucinogenic component to actually trigger, if we need this intracellular pathway to trigger the hallucinogenic compound, what happens if, for example, we make serotonin go inside the cell? And that what they show is that it had the same effect. So there is something very particular here that we still are trying to understand. And one of the things that we don't know, it's the one that you pointed out, that is like why there are some 5-HT2A agonists that are non-hallucinogenic and why there's others that are hallucinogenics, and what are they doing and why they are triggering these different pathways. So I don't think we have the exact answer. Also because we as preclinical scientists, what we do is, okay, we are going to bias something to a pathway, really through a pathway, and then we try to understand what's happening. But for example, LSD is just a partial agonist. So even if we do studies to try to understand what is the component of full agonists of 5-HT2A, maybe it's not what it's doing. So I'm sorry not to give you like a clear answer, but hopefully in the next years with some more research, we will be able to understand more about that.
Yeah, if you're taking part in the conference, I don't know if you are, like tomorrow David Olson will talk about, and that's his paper. So you can find out more from him directly. So he will be part of the symposium.
Hey, at risk of a maybe inflammatory question, because I know a lot of people like therapy, given these drugs seem to work in animal models, we're not giving the animal models talk therapy. Does this suggest that the, I guess, true psychedelics, in contrast with the entactogen MDMA scene, that we might be barking up the wrong tree if we're trying to, I guess, follow the MDMA pathway of this enabling talk therapy when it seems like the drugs are working pretty well by themselves? Do you have any thought on, I guess, what the animal functionality suggests for the future of human therapy?
Well, they are really big questions, guys. I'm just starting my PhD, but I don't think we are going to crack the field now, but I like to talk and to think with you about this subject. So one of the main problems also is that, okay, we have a lot of information on the acute effect, right? We know everything that is happening at the acute effect. What about the long term? You know, like what we are interested is not to trigger some plasticity or to trigger some behaviors at the acute level. And research has been quite difficult to show that at the long term. And most of the studies, what's happening, it's because animals, they don't have the same 5-HT2A receptors as in humans, for example. So they have this dissociation rate that is very, very fast. And so what we need to do, or what some I want to say colleagues, they are not my colleagues, but you know, some people in Canada, for example, from the Gobi Lab, the Gregorio, Antonio Inserra, what they are doing is that they are administering LSD daily, and then they can see some effects at long term, but up to a week, 2 weeks. So we are still struggling to tackle these questions about the long-term effects in animal research. So I would not jump to the conclusion of like, okay, they seem to be the holy grail. We should, why shouldn't we research other drugs? And on top of that, also to stress out to all the clinicians and to all the people just interested in the field, the more we know, the less we know, but the better. Because we also need to acknowledge that research and knowledge per se, even if it doesn't go to the clinical side, is important.
Yeah, and if I may add, so I think the debate is still open if self-experience is important and therapeutic intervention. It is not settled. I think there is strong evidence on both sides, so I guess there will be talks and panels during the whole conference, which probably will bring you a bit closer to your question. And but it's not settled, yeah, if this self-experience is important, because with the coming in of DMT, which is also healing, which is very, very short. And now we ask the question like, is a 10-minute self-experience healing? And then what about like something like LSD, which is over 8 hours? It's open. The verdict is open. Yeah, it's not settled yet. So last question.
Maybe in the same direction. Really nice talk and really is good summary. Really cool. The last slide you basically talked about, which we just talked about, is kind of the problem, in my opinion, of the field, the translatability between human studies and animal studies. The way I see it, maybe we're using the wrong animal models. And I'm asking you this question. Is what we find out in the animal models really that well translatable to humans, at least depression models, anhedonia models, PTSD models, and so on? Or do we need to take a different approach to actually find out how we translate from humans to animals?
Thank you. So I have now the responsibility of talking in name of all the preclinical fields. So one thing that I want to stress out is that all these experiments that you are seeing, we can do it, and we do it in animals, especially in rodents, because we have developed over the years exceptional techniques that allow us to be precise and causal, something that we cannot do in the human field. For example, the paper that I briefly presented that is on bioRxiv, we can actually tackle the question, is context important or no? Because we can put the animal, give him, for example, psilocybin or any other psychedelic drug, put him in a different context and see how it reacts. Put him in a stressed context, put him in a good enrichment environment. So we can tackle all these questions with the animals. And on top of that, at the neuronal level, we have circuits that allow us to, as we saw in the first talks, we go to a specifically brain region, to a specific neuron, and now you say you're active, you're inactive. And you see what that specific manipulation is going to do, to do to the behavioral level, to the neural level. So I don't think we should either do clinical research or preclinical research. I think we really need to work together if we want these medicines to become a standard in the field or not, depending on what we actually find out. That's my take-home message. So, yeah, thank you, guys.